Hello everyone and welcome to CBL. So today we'll be having a look at the current affairs in science and technology from 8th of December to 15th of December. So let's begin and see what are the major issues which we will be covering. The first important thing which we'll discuss today is another test for the coronavirus novel COVID-19. We'll have look at one of the major antibody tests that has been designed by the University of California. The next issue which we will be covering is China moon probe heads back to Earth. Another achievement for China if the probe heads back to Earth today, December 15th. We'll look into this China probe also. The next important thing which we will be discussing is SpaceX's Starship prototype explodes on landing after test launch. After a successful test launch, SpaceX's prototype has exploded. So we'll see what is the cause behind this. Next important issue which we'll be covering is Indian-American Rajachari among the 18 astronauts that will be in the NASA's manned moon mission. A proud moment for all the Indians. Again, an Indian origin Rajachari will be going to moon in 2024. Next important thing which is to be discussed is emergency use authorization of COVID-19 vaccine. All of us know the pandemic is still not over and the various pharmaceutical companies are trying to get an emergency use authorization. So what is it all about? We'll discuss in a while. So let's move on to the very first thing which we'll discuss today. That is obviously the coronavirus COVID-19 antibody test gives results in 20 minutes. As per the news, the results would be available in 20 minutes. University of California says that they have tried to combine the serological tests, that is, the test using the serum from human blood sample, the serological tests with the ELISA test. So they are trying to combine the fastness of the serological test along with the accuracy of ELISA test to get a new antibody test which is named as BELISA. B-L-I-S-A. And as per the University of California, this would be a better test as this would be able to detect low levels of antibodies and having a high specificity. So before moving ahead, let me tell you what is this ELISA test. ELISA test is done for a number of viruses, be it the AIDS virus or the rotavirus or certain malarial infections or dengue infections as well. So ELISA is a very accurate test and it stands for enzyme-linked immunosorbent assay. E for enzyme, linked immunosorbent assay. So this is a highly accurate and precise measurement which gives us the qualitative as well as the quantitative aspect of the virus or the pathogen which is to be studied. So it involves both qualitative and quantitative analysis of the antigen or the antibody. I would again like to tell you quantitative analysis is much more important because it would help our researchers to design the COVID vaccine. What amount of antibodies should be added into the vaccine so that it produces a desired result. That is why they have started with that BELISA test, B-L-I-S-A. Coming back to ELISA test, it is of various types. Let me tell you the names of those. Direct ELISA test, indirect ELISA test, then we have got competitive ELISA test and the sandwich ELISA test. But we are not going to study all these tests. I'm just going to tell you what is the basic underlying technique behind ELISA. For the ELISA test, we take a 96 well microtiter plate which already has the antigen which is to be considered, the antigen which we want to study. So, after having this microtiter plate, we add a blocking solution, block solution. So that block solution is generally BSA, that is bovine serum albumin, which helps to block the empty part of that well so that no further antibody can attach on any part of the well. And obviously that would give a better and accurate measure quantitatively. After this, we wash it with a buffer solution, and then into this well we add the serological sample. Once the sample is added, the sample which is to be tested, if I say about corona or any other virus, so the serum of that particular blood sample of the patient might be having the antibodies if he or she has the particular infection. So once the sample is added, the antibodies are going to attach themselves as they are very specific to these antigens. Right. Moving on to the next step, this is the primary antibody from the sample. Now again we wash the sample, and into that well we add the secondary antibody. The secondary antibody is very specific to the primary antibody present in the sample of that patient. So it attaches to that, the secondary antibody attaches to that primary antibody, and it is enzyme linked. Right, from here the name comes enzyme linked, because the secondary antibody that we have added is already enzyme linked, and to that specific enzyme we add the substrate. On adding the substrate, we get a colored solution, and if it happens, this shows that the person whose sample is to be tested, whose sample is being tested here, is positive for that particular infection. Because if he or she would not have been positive, there would not have been any interaction between the antibody, the secondary antibody and the primary antibody. And obviously if those antibodies are present and they are interacting with that antigen present, the person is said to be positive for that particular infection. I hope I've made myself clear what is this ELISA test is all about. So basically, by this ELISA test which the University of California has suggested, they want to say that we can have a better quantitative test and we can get an idea as to how much amount of level of a particular antibodies must be added to provide protection against infection with coronavirus. So ELISA basically stands for biolayer interferometry. Now this uses an optical fiber biosensor tip. Don't worry about this concept, fiber optic biosensor. Just you have to remember that this is a qualitative and a quantitative test which is of a short duration and much more accurate. So once this fiber optic biosensor is there, this sample can be analyzed by interferometry. And the process conducted is called as dip and read format, an automated one obviously. So dip and read format, what happens in this dip and read format? In the very first step, the biosensor tip is dipped into a solution containing the antigen. Now the tip already has the antigen, and after that, as the antigen binds to the biosensor, it is dipped into a wash solution so that extra antigen is removed, and it is dipped into the blood plasma sample, generating a signal as antibodies bind to the antigen. Once the antigen binds to the antibodies, a biochemical signal is generated which can be analyzed by the interferometer. So this makes the test much more fast and much more easier, giving the results in just 20 minutes. So again, a very important test after the RT-PCR, RT-LAMP and other serological tests. This BELISA test by University of California would help the scientists to obtain the vaccine and understand what level of antibodies should be added to get the desired response. Right. So now I move on to the next issue, which is China moon probe heads back to Earth. Now, Chang'e 5 probe launched on November 24th was a major venture by the Chinese Space Agency. So now they have already been successful, and now this probe is returning back to Earth, expected on December 15, that is today. It was a 23-day long venture where the probe is supposed to bring about 2 kilograms of moon dust and samples back to Earth for the study. And China would become the third country after US and former Soviet Union to do that. So when it heads back to the Earth, then only we'll get to know whether the 2 kilogram sample has been collected and what are the implications, what are the various findings that have been concluded from this probe. Basically, this Chang'e 5 probe was to land on the Mons Runker region on the moon, which has not been visited previously. So an important mission where this probe is coming back to Earth and will bring the rock samples for the study. Right. Now I move on to the next issue. The next issue which is very important is SpaceX's Starship prototype explodes on landing after test launch. After the successful test launch of Starship, which is supposed to be the biggest cargo and is supposed to carry the cargo and the humans in NASA's other missions, lunar missions and Mars missions. After a successful test launch, while landing, there was an explosion and it exploded, right? The reason behind this was that while descending, the velocity was much more higher than what was required. This rocket, the Starship rocket, was destroyed. It was about 16-story tall prototype to lift the heavy launch vehicle developed by billionaire Elon Musk. So according to Elon Musk, as per the return journey, when this Starship was returning, then there was a massive fuel header tank pressure low. This low pressure led to the velocity which was much more higher than what was expected. So low pressure and this high velocity resulted in the explosion. A setback, but obviously Starship is a much more bigger prototype and it can be reconstructed, and then NASA's manned mission would be carried on with this Starship. Next important issue which we are going to discuss is Indian-American Rajachari among 18 astronauts selected for NASA's manned moon mission. 18 astronauts, 9 females and 9 males have been selected for the NASA's manned mission in 2024. This mission would be carrying the first woman to the moon surface, and among them, proud moment for India, the Indian origin Rajachari, who is an Indian-American US Air Force colonel, would be into that mission. This Artemis mission, Artemis team, what is it going to study? It is going to study the basic lunar, the major important lunar prototypes and the lunar volatiles over there, and gathering more and more knowledge by this Artemis mission about the planetary process, the origin of Earth and the relationship between Earth and Moon. So let us have a look what is this Artemis mission about, and Rajachari would be a part of this Artemis 3 mission. It will help us to understand planetary processes, understand the character and origin of lunar polar volatiles, revealing the record of ancient sun and other astronomical environment, and then observing the universe and other planetary things from a different, unique location. Obviously, they'll be conducting a number of experiments which would be helpful. So, Rajachari would be a part of this Artemis 3 mission. Right, moving on to the next thing. is the fourth person that would of Indian origin that will be going to the moon. And the other three were, you must remember the names, Rakesh Sharma, April 3rd, 1984. He flew aboard the Soviet Soyuz T-11. And then there was Kalpana Chawla, all of us know, in the ill-fated Columbia Space Shuttle while returning back to Earth on February 1, 2003, exploded, killing all the crew members. And Kalpana Chawla was a part of that mission. Right. After that, we would say Sunita Williams, a very eminent name, a very eminent cosmonaut, the only female cosmonaut. She has lost more than 321 days in space and having a walk of 50 hours and 40 minutes in her 7 excursions to the space. So, an eminent women space traveler, again an Indian. And the fourth one would be Rajachari. Right. Now, I'll move on to the next issue, that is, can vaccines with emergency use approval be used at the population level? Again, a big question mark. All of us are suffering from this pandemic and we want to have the vaccine as soon as possible. So, a number of pharmaceutical companies are trying to attain the approval by the Central Drug Standard Control Organization for using the COVID vaccine. It has already been started in US, UK and Canada. As for today's news, Canada has also started it. So, a number of vaccines but is it okay to begin with these emergency use? Because some of the vaccines are still into their phase trials, be it phase 2 or phase 3 trials. And the data is still not sufficient as to should this vaccine be used for the populations. The different vaccines that are vaccine candidates that are trying to get the approval are COVID Shield, which is manufactured by SII Pune with the help of Oxford University and AstraZeneca. This particular vaccine is like very important for India and they are into the phase 3 trials. The same vaccine has been used in UK and Brazil. They have already started it, but we are still waiting for the results of the phase 3 trials. And as soon as those analysis is done about the third trial, so this vaccine would be available to all of us. The next important vaccine is Covaxin, again manufactured by Bharat Biotech with ICMR, that is ICMR's National Institute of Virology. The question is again the same. They have started the phase 3 trials and only the results are still awaited. We are not sure what would be the results. But if I talk about the other Pfizer vaccines, BN2-162b2, they've already started giving vaccines in US and UK. In UK, they've administered it to 91-year-old female and then there was 81-year-old male. And recently, the US nurses have already got that vaccine and they are feeling much more better. So, the trials are still awaited, clinical trials are still going on, but the emergency situation has forced CDSCO to give the approvals even if they are into the third trials and the results are still awaited. Next, I am going to give you a few things, food for thought for today. Like you have to study yourself what is the great conjunction of Jupiter and Saturn, and I'll discuss it in the upcoming lecture. Let me give you a small hint which we'll be discussing in the next lecture. This great conjunction of two giant planets, Jupiter and Saturn, is going to occur on 21st of December 2020. Do let me know about it and we'll discuss it in the next lecture. Let's see what is this great conjunction is all about. All of you must be aware of the Nobel laureates, and Nobel Prize 2020 awards function has already been concluded on 10th of December on a digital platform where the diplomas and the medals were given to the laureates at their hometowns. So you must go through all those laureates and the important information about it. We will be discussing these two in the next lecture, but this is your food for thought. You have to study it and we'll discuss it. Until then, bye from my side. Thank you.